And now we're going to hear from George McKee. She's the program officer for cornea and inflammation and infection.
I'd like to thank everybody today for inviting me to speak about the cornea portfolio. And I just want to emphasize the cornea is not just the dust cover. I oftentimes hear, oh, it's just the dust cover, don't worry about that. And I'm sure other cornea researchers do as well. And we're going to emphasize, we're going to try and convince you, I'm going to try and convince you to emphasize the importance of the cornea. So if you'll bear with me for just a minute, I just want to get everybody in the room kind of on the same page with the location description of the cornea. The cornea is in, let me turn my mouse here so I can drive right and go the right direction. Hold on just a second, technical difficulties. The cornea is this, the tissue on the front part of the eye, that's clear, dome-shaped tissue, and you can see the iris, which is the colored tissue you look through the cornea to see, and the pupil is a black hole in the middle of this tissue. So the cornea is this clear outer tissue that covers this part of the eye. And histologically, there's the epithelium, which is a non-keratinized stratified epithelium on the front surface, similar to your skin. Bowman's layer, the main thickness of the cornea is stroma, fibroblast, collagen, and on the posterior part of the cornea is Descemet's membrane, which is the basement membrane to a thin layer of cells called the endothelium. And the endothelium is just a single layer of cells that you're born with. They don't regenerate. Once they die off, you're out of luck. So to give you an idea, the relative size of the cornea program, we're the third largest program in the NEI, second to, or third to retinal diseases and SAVP. So we have about 13% of the NEI applications that come in and funded are in the cornea program. This slide is just to give you an overview of the research being done in the portfolio. And this, by all means, does not cover everything because there's a broad depth of investigation in many areas in the cornea portfolio. And so I just kind of listed the topics that have the most applications in these areas. But again, there's many, many areas that aren't listed here. So this isn't by any means the full depth of the portfolio. And I listed investigators on here. And again, these are investigators. I'm just going to highlight some work they've done in each area just to give you kind of an idea, but again, it's not the full depth of the work being done here. So a large majority of the portfolio does involve dry eye and tears, which you'll see are very important to the maintenance of the cornea. And Dr. Gordon Laurie, his latest research is he's looking at lacritrin, which is a normal component of tears, and he's very close to studying a lacritrin replacement drop for people with dry eye, believing that it's really essential for normal tearing and for ocular surface maintenance. Infectious diseases of the cornea is probably the largest component of my portfolio. And there's many components of infectious diseases, viral, bacterial, fungal, viral being herpes, as you guys know, since I'm generally presenting biohazards for herpes. And I listed 3 investigators, and these are 3 different independent labs, Dr. Carr, Gassi, and bin Mohammed. They're all looking at development of a herpes vaccine and are probably going to be pretty close to getting a herpes vaccine out there in the next few years. Adenovirus, which you guys are all familiar with, if your kids get conjunctivitis, they get to spend some days at home. Dr. Chodish is a researcher in that area. Bacteria, Pseudomonas and Staphylococcus. I'll mention Dr. Fleshig, but I'm going to talk about her work in a little bit more detail in my presentation. Fungal infections, Dr. Perlman, and he's looking at the creation of an alternative to steroids for the treatment of fungal infections. So oftentimes after a fungal infection, the patient's put on topical steroids and they get a reoccurring fungal infection. So he's looking for an alternative to treat these patients so their immune system isn't suppressed and they don't get reoccurring infections. Stem cells, as you can imagine, is a very big area in the cornea right now because there are stem cells and they do regenerate. And a lot of the work recently has been taking the stem cells and transplanting them for wound healing and finding out where they're actually located and how they differentiate. There's been some recent findings thinking they're not always located in the limbus. They're located throughout the cornea. So that's been interesting. Endothelial disease. The most common endothelial disease is Fuchs endothelial dystrophy. Doctors Afshari, Ghosh, and Mutha are looking at the genetics of Fuchs endothelial dystrophy. And I'm going to back up for a minute. I just realized I skipped wound healing. Wound healing is another big part of the portfolio, as you can imagine. And Dr. Murphy is looking at the mechanical forces of the environment on the epithelial cells, not just the chemical, but the mechanical forces and their effects. Corneal transplants, Dr. Shui, and refraction, Dr. Zhang, I'm going to set them aside for a minute because I'm going to go into more detail about their work later on in this presentation. Structural malformations, I wasn't really quite sure where, that was a term I created for keratoconus because it's bulging of the cornea, and I'm not really sure where you would stick it. But we've had a lot of genetics works done with keratoconus too, with Dr. Rabinowitz and Chakravarty. So again, I'm not going to talk about each of those aspects of the portfolio because there's way too much to cover. So I narrow it down to what I like to call the 3 superpowers of the cornea. The cornea is a barrier of protection, helps shield the eye from the environment. And it's also the most refractive tissue in the eye. The overall refraction of the eye is about 60 diopters. The cornea has 40 of those, roughly around 40 of those diopters, whereas the lens just has a third of that, around 20 or so. It's also the most highly innervated tissue in the body, so it has 300 to 600 times the sensitivity of the skin. The cornea is really good at fighting back. I'm going to start with, let me back up and say I'm going to start with the first superpower of the cornea. The cornea is pretty good at being resistant to infections, and I'm leaning on infections as far as a barrier, something because that's the most common research being done in the area. And you can see here that it has really good defenses. If you put bacteria on an eye over time at 14 hours, almost all the bacteria, all the bacteria are gone. And this has been shown for both Pseudomonas aeruginosa and Staphylococcus aureus. Now, Dr. Suzanne Fleshig, who is studying bacterial infections of the cornea, and she studies contact lens-related bacterial infections, since contact lens-related bacterial infections is the most common cause of bacterial infections in the cornea is wearing a contact lens. And it's the most common cause of blindness due to corneal disease is contact lens-related bacterial disease. And so since she wants to look at how a healthy cornea resists bacteria versus how a sick cornea reacts to this bacteria. So she's looking at whole healthy eyes. So this is an example of an imaging technology she created, and this is of a mouse cornea that's not been sectioned, it's not been fixed, it's a live mouse, and she's taken Pseudomonas and put on this cornea, and she picked Pseudomonas since it's the most common contact lens related infection. And you can see that she had to, this is after 6 hours, she put 1 billion colony forming units of Pseudomonas on this eye. And to get this bacteria to stick to the surface, she had to superficially abrade it with tissue blotting paper. Otherwise, the bacteria just fell off. And the only way it would and you can see it hasn't penetrated down to the stroma yet. The only way you can get it to penetrate the stroma is that actually injure the stroma. Now, investigators for a long time have been studying why is the cornea so resistant to bacteria, and 3 of the reasons they've discovered is the tear fluid. The tear fluid is very good at working on the microbes directly itself, but it also changes the gene expression in the corneal epithelial cells to make them more resistant to bacterial infections. And there's also been the discovery antimicrobial peptides are found in the corneal epithelial cells, and antimicrobial peptides are small keratin proteins within the epithelial cells act directly on the infections or pathogens themselves. They're kind of like little natural antibiotics if you think about it that way. And corneal epithelial cells also act as phagocytes, so they trap lysosomes inside. They trap bacteria inside their lysosomes where they kill them. You can see in this movie lysosomes filled with bacteria here. Now, unfortunately, Pseudomonas has capacity to avoid some of these corneal epithelial cells' natural defenses. And again, Dr. Fleshig's lab has discovered that Pseudomonas for a very long time was always considered to be extracellular Dr. Fleshig has discovered that there's a genotype of Pseudomonas that's actually intracellular. And you can see this is an image where Pseudomonas is circling around in this epithelial cell. It's replicating. It takes us and pushes the cytoplasm away from the plasma membrane and creates these little blebs where it replicates and creates a nice little happy, healthy home there. And it will prevent the cell from committing suicide so that it can live in its little happy home. The other thing that Pseudomonas has done is that to avoid being eaten by lysosomes, it blows the cell up before it's eaten, or let me show you this little video here. In these little blebs it creates, it gets inside these little blebs and uses it to transport to another corneal epithelial cell to invade. And in this slide, there's been 100 times the concentration of gentamicin put on this epithelium. And you can see the Pseudomonas is pretty well protected from its environment inside this little bleb. Now, to make matters worse, they've discovered that Pseudomonas adapts to the ocular surface over time to become even more virulent. So they've taken normal rats, done nothing to their eyes whatsoever, not abrade them, not scratch them or anything, and infected contact lenses with Pseudomonas. And they put the contact lenses on the rats, and in a median of about 7 days, all the rats get infectious keratitis. Now, if they take these contact lenses off those rats and put on other normal, healthy rats, the median time to infection goes from 7 days down to 2 days. So these rats are getting infected quicker. And they've noticed that once the Pseudomonas has been on the ocular surface for some period of time, they get a change in gene expression that allows them to invade corneal epithelial cells faster. All right, well, I'm going to leave that alone for a minute, leave the barrier alone, and we're going to go to the second superpower, which is the refractive error of the cornea, or the refraction of the cornea, correction of the refractive error. Because unfortunately, not all of us are born with eyes with normal refractive capabilities, and so for a very long time, many investigators have found a way to correct the corneal refraction to help improve our vision. The first eyeglasses were created in the 13th century in Italy, and I really tried hard to get a copy of these Italian eyeglasses. I thought they'd be kind of cool, but I couldn't find them. The first contact lenses, or not a copy of the actual glasses, but a picture. The first contact lenses were made out of glass in 1988 in Switzerland. And in 1970s, excimer laser was developed, and Dr. Trocco from Columbia University was the first person to use this laser on rabbit and monkey, on cadavers, and then live rabbit and monkey corneas. And in 1988, a 60-year-old woman volunteered to be the first person to have a photorefractive keratotomy done. She had malignant melanoma and the eye was being removed, so she agreed to have this procedure performed. So she had the first PRK done, and this was done by Dr. Trocco's colleague, Dr. Margaret MacDonald. Then in the 90s, 2 European doctors discovered how to create a corneal flap instead of working just on the corneal surface, so PRK was turned into what's now known as LASIK surgery. And in 1999, LASIK was approved for use in the United States. In the 2000s, wavefront analysis was developed, which allowed more precise reshaping of the cornea based on the individual patient. And to cut the blade went to using a femtosecond. To cut the flap they used a femtosecond laser to cut the flap instead of a blade. Now, I look around the room and not many of you are old enough to understand presbyopia yet, but trust me, you all will get there. And I like to say there are many stages of presbyopia. You have the denial phase where you do this. Oh yeah, I can see. Then you have the acceptance phase, and you shop for the pair of glasses that are very fashionable. You want the right color, the right shape for your face. And then you get to the phase where it doesn't matter anymore. You just buy glasses and leave them all in every room of your house, in every purse. You have always a pair with you. Now, presbyopia doesn't affect the cornea directly, but an engineer at the University of Wisconsin has decided that he's going to help the cornea correct the refraction for what the lens can't do on its own. And he's 38 years old, and he got an NIH Director's New Innovator Award. And he states, I'm 38, and I want to be the first person to use this contact lens when I'm in my 40s because I don't want to have glasses. And he, being an engineer, has three very challenging engineer components to this contact lens. The center of this lens is a tunable liquid lens. The center is 6 millimeters, so it covers your pupil, and it's made from a silicone water interface that is a thermosensitive hydrogel that changes shape based on this thermosensitive hydrogel. And then around this tunable liquid lens, he's putting sensors and control electronics for autofocusing like a camera. Dr. Zhang realizes he's got to have a power source to supply power to these sensors and control electronics. And he knows that you can't even put the smallest battery in a contact lens. So he's going to harvest energy from the environment. So around the periphery of the contact lens, he's going to put other small engineering components consisting of small silicone nanowires and polymers, a lot of engineering stuff I don't really understand. And he's going to develop these little components so that it not only harvests energy from the environment, but it stores them so this contact lens is functional for a couple hours when the environmental source is weak or even gone for these contact lenses. And the third challenge for this engineer is getting all these components molded to a contact lens, a flexible material. And he plans on using contact lenses that are already available so that there's patient safety and comfort out there. And he's going to create a special contact lens for you to put your contacts when you take them out so that you store them overnight with this lighted system so that when you put them in the next day, they're fully charged and ready to go. And he is in his last year of his award, and he has created, according to his reports to us, he's created all the components, and he's getting ready to put all of these into a contact lens. So cross your fingers. We'll see where this goes. Now I'm going to switch to the third superpower, which is the corneal nerves. The corneal nerves are supplied by the peripheral and central trigeminal network, and they perceive mechanical, thermal, and chemical stimuli and elicit pain responses. So you withdraw from this stimuli. And they also work to maintain the optical tear film and produce trophic factors to maintain a healthy state of the cornea. Unfortunately, I feel that we have very few investigators involving corneal nerves right now in the portfolio. The investigators we do have involving corneal nerves are looking at abnormal dry eye sensations associated with people who have had dry eye that's responding to treatment, or maybe it's resolved and they still have these dry eye sensations in their eyes and they're wondering why these nerves continuously fire when stimulated. And so this actually is an area of the portfolio that I think we could use some more, the superpower could use some more help. Now I'm going to switch topics all over again and talk about corneal transplants, because I don't know about cornea researchers in general, but I used to hear, oh, you can just transplant the cornea, what matters? You know, you can take care of it and put a new one in. And in 1905, but fortunately for cornea researchers, yes, we can transplant the cornea, but that's not 100% a sure thing for everybody yet. In 1905, the first successful human corneal transplant was performed, and you can see on the side it was after many decades of trial and error, from transplantation in a gazelle to using a xenograft, which is from a different species in a human, which not surprisingly, that wasn't successful. And it wasn't all this, Dr. Zierum's success in 1905 came along finally with antiseptic and anesthesiology increases. But it wasn't until the way it is more routine today didn't occur until advancements in immunology, the development of topical corticosteroids, antibiotics, and advances in surgical equipment such as the tree find, the operating microscope, suture material, and so forth. Now I'll take a minute and kind of give you a brief diagram, and this really simplifies corneal transplant surgery. I'm sure Dr. McLeod and Dr. Weiss are cringing because this makes it way too simple. But you have a patient in here, and he's got his lids open with the retractor, and there's a tree find that goes in and punches out the cornea. It's like a cookie cutter, punches out the cornea you want to remove, and you then take and cut out the piece of the cornea, or the piece of the section of the cornea you want to replace. You put the donor cornea in because it always fits right in there just perfectly like it was made to, and then you meticulously suture this cornea in. And I can tell you, having tried this, that this takes a lot of practice getting those sutures meticulously put in there. So today, corneal transplants are successful, I would say 80–90% of the time, but there are still complications associated with corneal transplants. There's eye infections, cataracts, glaucoma, vision problems, anywhere from not having a normal refraction because you're taking someone else's cornea and putting in the refractive capability of your eye, which really doesn't mess up. I'd say the most common complication is rejection, and that occurs about 20% of the time, roughly. And despite that, though, even with patients who have had rejection, it can be 10 to 20 years, so you can live for a long time with a corneal transplant. But the concern about corneal transplants now is the availability of a cornea for patients who need them. So one of the two reasons that they're less available is an aging population. Now, it's been shown that it doesn't really matter how old the cornea is that you use, as long as the endothelial cells, which is the single layer on the back part of your eye, they are high enough in number because they control the dehydration of the cornea and keep it clear. And as you age, you lose your endothelial cell count. So you have to be able to catch those endothelial cells. And the other concern is many people are having LASIK procedures or other refractive surgeries now, which makes that cornea no longer available for transplantation. So due to this, there are artificial corneas on the market. So people have looked at ways to not use a human cornea to a human, but take and create artificial cornea like out of polymethyl methacrylate. And the Boston K-Pro is probably the most commonly used one. It's been on the market for over 200 years. But the problem with the Boston K-Pro is that you're required to have a donor cornea or a corneal transplant to suture the prosthetic into, and then you take that and you put that into your host cornea. So now you have all the complications of the cornea transplant plus the additional complications of the artificial cornea, which include extrusion and infection. Now, the Alpha Core is the other one on the market, and it's nice because you don't have to have a cornea to insert it into. You can just insert it into your host eye. But it does require a pocket that takes about a meticulous 3-hour period of time to dissect this pocket to put this prosthetic into. And it has a different set of complications. There's still the added risk of extrusion and infection, but it takes about 3 months for this pocket to heal, and during this time, this patient is not able to see out of this eye while you're waiting for this pocket to heal. Now, Dr. Shui is a NAEC researcher that has recently worked on developing what's called the CARACLEAR. The CARACLEAR is approved for use in Europe and is awaiting FDA trial here in the United States before human trials are started. And you can see the cornea on the left needs a transplant, and there's a picture on the right where this CARACLEAR has been placed in this patient. And the nice thing about the CARACLEAR is it's made of a flexible polymer. So you can just insert it, you can just pop it into the cornea, into a partial thickness with a femtosecond laser through a 3.5 millimeter incision. So the time to do the surgery decreases to around 15 minutes versus several hours with corneal transplants or other prosthetics. And the other nice thing about this prosthetic is there's less chance of infection. You have immediate visual recovery. And according to Dr. Shui, you can control astigmatism and refractive error with this polymer as well. So I hope that you may have been thinking through this talk that, oh yeah, it's really just a dust cover. And I must say that when I wrote this talk, I was kind of thinking, yeah, it kind of is. But I hope I leave you with the impression that it is a dust cover, but it's got a lot of very impressive capabilities with it.